this video, I'm going to talk about the concept of Krebs cycle. Krebs cycle is also called as citric acid cycle or tricarboxylic acid cycle or a TCA cycle. It is called as Krebs cycle because this cycle or series of reaction is discovered by English biochemist named Hans Krebs. That's why it is called as Krebs cycle, or this cycle named after the discoverer Hans Krebs. This Krebs cycle is also called as a citric acid cycle because in this series of reaction, the first formed compound is a citrate or a citric acid. That's why Krebs cycle is also called as a citric acid cycle. The citric acid cycle is also called as a tricarboxylic acid cycle or a TCA cycle because in this series of reaction, most of the compound that is formed or the intermediate compound that is formed consisting of mainly 3 carboxyl group or 3 COOH group. That's why this cycle is also called as tricarboxylic acid cycle or a TCA cycle. So these are the different names of a Krebs cycle. Similar to that of glycolysis, even Krebs cycle is a main step of a cellular aerobic respiration. Though it is a part of aerobic respiration, the whole process of Krebs cycle does not require not even a single molecule of oxygen, or in any step of the Krebs cycle, it does not consume not even a single molecule of oxygen. Where does this Krebs cycle takes place then? Krebs cycle takes place in the mitochondrial matrix, and all the enzyme that is essential for the Krebs cycle is dissolved in the mitochondrial matrix itself. Now let me talk about the different steps of the Krebs cycle and different enzyme involved in the Krebs cycle. The Krebs cycle begins with the 2-carbon compound called as acetyl coenzyme A. This acetyl coenzyme A is formed by the pyruvate, which is the final product of the glycolysis. As I discussed in the previous video, the pyruvate, that is 2 molecule of pyruvate which is formed as a result of glycolysis, undergo oxidative decarboxylation to produce 2-carbon compound called acetyl coenzyme A. This 2-carbon acetyl coenzyme A reacts with the 4-carbon compound called as oxaloacetate to form a 6-carbon compound called as citrate or citric acid, which is the first step of the Krebs cycle. So what happens in the first step? Acetyl coenzyme A reacts or combines with the oxaloacetate to produce citrate or a citric acid in the presence of water. So in the presence of water, it produces citrate or a citric acid. During this reaction, CoA, that is coenzyme A of acetyl coenzyme A, is released, and the enzyme which catalyzes this reaction is citrate synthetase. So in the first step, the citrate or citric acid is produced, which is a 6-carbon compound. In the next step, this citrate or citric acid, which is a 6-carbon compound, loses a molecule of water to produce cis-aconitate. So, citrate loses a molecule of water to produce cis-aconitate, and this reaction is catalyzed by the enzyme known as aconitase. Later, this cis-aconitate undergoes certain isomeric changes to produce isocitrate, or cis-aconitate in the presence of water molecule undergo isomeric rearrangement to produce isocitrate, or cis-aconitate changes into its isomer known as isocitrate in the presence of water molecule, and this reaction is catalyzed by the enzyme known as aconitase. So this isocitrate is again a 6-carbon compound. Later, this isocitrate get oxidized or undergo oxidation process to produce oxalosuccinate, which is again a 6-carbon compound. During this oxidation process, 2 hydrogen atom is released, and these 2 hydrogen atom is received by NAD+, that is nicotinamide adenine dinucleotide. So after receiving 2 hydrogen atom, this NAD+ reduced to NADH and H+. So isocitrate is oxidized to oxalosuccinate, and NAD+ is reduced to NADH and H+. This reaction is catalyzed by the enzyme known as isocitric dehydrogenase. So ultimately 6-carbon compound, that is oxalosuccinate, is produced. This oxalosuccinate, which is a 6-carbon compound, loses 1 carbon in the form of carbon dioxide to produce alpha-ketoglutarate. So this alpha-ketoglutarate is a 5 carbon because the oxalosuccinate loses 1 carbon in the form of carbon dioxide to produce 5 carbon compound, that is alpha ketoglutarate. In the next step, this 5 carbon compound, that is alpha ketoglutarate, get oxidized or undergo oxidation with the removal of carbon dioxide to produce 4 carbon compound called as succinyl CoA. So 5 carbon alpha ketoglutarate remove 1 carbon or loses 1 carbon in the form of carbon dioxide to produce 4 carbon compound called as succinyl CoA, and losing of carbon dioxide takes place along with the oxidation of alpha ketoglutarate. During oxidation, 2 hydrogen atom is released and this hydrogen atom is received by NAD+. So after receiving 2 hydrogen atom, NAD+ is reduced to NADH and H+. So alpha ketoglutarate is oxidized to succinyl CoA, NAD+ is reduced to NADH and H+. This reaction is catalyzed by the enzyme known as alpha ketoglutarate dehydrogenase, which produces finally succinyl CoA. In the next step, this succinyl CoA, which is a 4 carbon compound, is formed or converted into simple compound known as succinate with the removal of coenzyme A. So in the presence of water, succinyl CoA is converted into succinate with the removal of coenzyme A or CoA. So along with the release of coenzyme A, large quantity of energy is released, and this energy is utilized to add phosphate group to GDP. GDP means guanine diphosphate. So one phosphate is added to GDP, which convert GDP into GTP. Anyhow, later this GTP gives or donates the phosphate group to ADP. So adenine diphosphate. So upon receiving phosphate, this ADP is converted into ATP, and this reaction is catalyzed by the enzyme known as succinyl CoA synthetase. In the next step, the simple compound, that is succinate, get oxidized or undergo oxidation to produce fumarate. So succinate is a 4 carbon compound which oxidized to produce fumarate. Fumarate is also 4 carbon compound. During oxidation, 2 hydrogen atom is removed, and this removed hydrogen atom is received by FAD, that is flavin adenine dinucleotide. So upon receiving 2 hydrogen atom, FAD is converted into FADH2, and this reaction is catalyzed by the enzyme known as succinic dehydrogenase. In the next step, this fumarate in the presence of water is converted into malate, or in the presence of water, fumarate is converted into malate, and this reaction is catalyzed by the enzyme known as fumarase. In the next step or in the last step, 2 hydrogen atom is removed from the malate, which convert malate into oxaloacetate, which is again a 4 carbon compound. The 2 hydrogen atom which is released from the malate is received by NAD+. So upon receiving 2 hydrogen atom, NAD+ is reduced to NADH and H+. So this reaction is catalyzed by the enzyme known as malic dehydrogenase. So in the presence of malic dehydrogenase, malate produces oxaloacetate, or malate is converted into oxaloacetate. So this oxaloacetate is ultimately cycled back. Later this oxaloacetate will combine with another acetyl coenzyme molecule to repeat the Krebs cycle or repeat this citric acid cycle. So ultimately at the end of the Krebs cycle, oxaloacetate is cycled back. So that is all about the different steps and the enzyme involved in the Krebs cycle or a citric acid cycle. I hope this video will be useful. If you like this video, please hit the like button and subscribe to my channel. Thank you.